Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor in IIT Delhi. Today we are going to talk on the module which is entitled Types of Immunity, Innate and Acquired Immunity. This is of course from the paper called Immunology. In this module, we will learn of the two types of immunity, namely innate and acquired. We will also learn about how the innate immunity functions and which cells are responsible for this kind of immunity. The objectives of this module are to understand the basic difference between innate and acquired immunity, to learn about various cells which are involved in innate immunity, thirdly to learn about various soluble factors which again are involved in innate immunity, because innate immunity functioning requires the involvement of the soluble factors as well as various cells. Lastly, we will also try to understand how innate immunity also has a limited specificity towards pathogen via pattern recognition. So, the concepts which will be covered will be essentially to learn about these two types of immunity which are called innate and acquired. In the innate immunity part, we will learn about phagocytes, natural killer cells, abbreviated normally just as NK cells, dendritic cells and mast cells. These all cells are part of the innate immunity. On the other hand, acquired immunity responses involve basically two types of lymphocytes. One is the B cells and the second one are the T cells. So, as we have already mentioned that there are two types of immunity, innate, with which we are sort of born with and which actually exist even before we encounter any infectious agent. The second one is the acquired immunity, which comes into the play when the encounter with the infectious agent takes place. Innate immunity and acquired immunity complement each other. The response of the innate immunity system is fast. It has some limited specificity for the microbial infectious agents. However, it has no memory. Every time the innate immune system encounters an antigen, it reacts ab initio. It doesn't remember that the same infectious agent was encountered. Hence, the innate immune response is not improved by repetitive invasion by the same antigen. These three features actually distinguishes it from acquired immunity. Acquired immunity is also called adaptive immunity. It is antibody mediated, and not only antibodies, the T cells also mediate the response of the in terms of the acquired immunity. All this will be discussed in more detail in later modules. This particular module today will focus just on innate immunity. Let us look at the factors which participate in the immune response. The cells responsible for the innate immune responses are phagocytes, natural killer cells, which we will normally call NKC in further lectures. Then there are mast cells, and finally there are dendritic cells. In other subsequent lectures, we will have an occasion to describe these cells in a greater detail. However, in this module, we will briefly look at the functioning of the various cells. Let us look at the functioning of the NKC as a cell which is involved in the functioning of the innate immunity. As you can see in this figure, the lower left, the NKC is locating, binding and attacking a large cancer cell. In the C part, the NKC has punctured and destroyed a large cancer cell. Finally, in the D part, what we see is that the tumor cell structural matrix is all that remains after its encounter with the NKC. Let us now look at the molecules responsible for the innate immune response. These are the so-called soluble factors which we talked about. Several classes of molecules participate in the innate immune response. These are antimicrobial peptides, which are abbreviated as AMPs, various pattern recognition receptors, abbreviated as TLRS. The prominent among these are toll-like receptors called TLRs and mannose receptors which are called MRS. The soluble pattern recognition receptors constitute in fact a larger list. We will look at it subsequently. Included in this soluble molecules which participate in the innate immune response, we have a very important system called complement system. Then there is a set of proteins called acute phase proteins, and finally last but not least, we have cytokines. First of all, let us look at the antimicrobial peptides or the AMPs as these are called. What is shown here are only some examples. This is not a comprehensive list. Epithelial cells of the respiratory, gastrointestinal and genitourinary tracts produce such antimicrobial peptides. If the microbial infection breaches the barrier of the epithelial cells, it encounters phagocytic cells. One of the modus operandi of phagocytic cells during innate immune response is in fact to produce some of these antimicrobial peptides. These peptides are not of very large length. They are generally made up of less than 60 amino acids. As far as their secondary structure is concerned, they may have a alpha helical structure, a beta sheet structure, an alpha beta mixed structure, or just a non alpha beta but extended structure. Let us look at the properties of antimicrobial peptides. AMPs cover a wide spectrum of antimicrobial properties. This range includes both gram-positive and gram-negative bacteria, viruses, fungi, many have net positive charge. Mechanism of action includes altering the membrane permeability of the cells of the pathogen. Some AMPs exploit chemotaxis and attract T cells and dendritic cells. Thus, AMPs are part of the cooperation between innate immunity and acquired immune system. Let us further look at some more examples of the AMPs. Specifically, we have defensins, which are listed here. Among the defensins, alpha defensin is secreted mostly by neutrophils. Sometimes epithelial cells also secrete them. Beta defensin is mostly secreted by epithelial cells and keratinocytes. Cathelicidins are mostly secreted by macrophages, Soracin by keratinocytes, and calprotection by PMNs. It is believed that we may not have yet discovered all antimicrobial peptides. However, this table shows a more exhaustive list of AMPs and their functions. The idea is to make you realize that the list is very large and we may even discover more of these in future. If you look at this list, we find that antimicrobial peptides can function as protease inhibitors. They can function as chemokines. They can also function as neuropeptides. If we look at this list, we find again the antimicrobial peptides which are secreted by keratinocytes. We had already mentioned cathelicidins and beta defensins, but in this lactoferrin and lysozyme can also be added. Lactoferrin and lysozyme and RNase 7 in fact are slightly bigger molecules and are normally more or less short globular proteins. Similarly, we have AMPs in infiltrating cells which are mentioned. Cathelicidins and alpha defensins again figure on this list, but we also have a perforin and platelet basic protein and fibrinopeptide A and B. Similarly, among the protease inhibitors, we have cystatin C, which is very important. As a chemokine, we had already mentioned soracin, but we already also have the three other peptides. Alpha MSH, bradykinin, neurotensin, all these are considered neuropeptides, and overall all of them come under the broad category of antimicrobial peptides. AMPs are either produced constitutively or produced via pathogen associated molecular patterns. The latter operates via pattern recognition receptors which are present on the macrophages, PMNs, myeloid and plasmacytoid dendritic cells, some epithelial cells. This mechanism is responsible for the limited specificity of the innate immune system. It results in removal of microbes with common or similar surface structural motifs through a single functional response. Apart from this, some soluble pattern recognition receptors also exist. All these receptors can also bind to self components and participate in apoptosis. Let us now move on to the various pattern recognition receptor. As we mentioned, an important example of this is a class which consists of or is generally called toll-like receptors. They are abbreviated normally as TLRs. Toll-like receptors are among the ancient receptors from the evolutionary perspective. That means as the life evolved in early days, the earlier organisms, when they evolved and the immune system was developing, toll-like receptors were among the early molecules which evolved in that era. Initially, toll receptor was discovered in the fruit fly Drosophila, and after the discovery in this organism, later various toll-like receptors were found in the mammalian systems. While most of the TLRs are present on the cell surface, especially on mononuclear phagocytes, some are also present on endosomes. Some of these terms which we are talking about, mononuclear phagocytes or endosomes, will become clearer when we formally talk about them in the right context. The idea here is to briefly indicate that the diversity of the cells on which these TLRs are present. Family of TLRs recognize wide range of pathogens. As you can see from the figure, TLRs are integral proteins having intracellular domains, which would mean that you have a part which is facing the cytosol, there is another part which is facing the outside of the cell, and as integral proteins by definition, they imply there is a domain of the TLR which is passing through the biomembrane. Let us briefly look at the function of the TLRs. How does these toll-like receptors function? What basically happens is that upon binding to the ligands on the pathogen, which would mean that they basically act as a receptor for these ligands which are present on the pathogen. Please remember that these are basically pattern recognition molecules, which would mean that they seek a pattern on the cell surface of the pathogen. The pattern is in terms of the distribution of the ligands on the cell surface of the pathogen. When this binding of the pathogen happens to the TLR, the cells containing TLRs produce cytokines. Cytokines are again soluble secretory molecules. Production of the cytokines has many consequences. For example, inflammatory response due to cytokines, which include TNF alpha and IL-2. TNF alpha and IL-2 are two important cytokines. As we further go into the details of the functioning of the what is known as a cell mediated immunity, we will have a more detailed look at some of these cytokines. Right now it suffices to say or to point out that these are the two cytokines which are part of the inflammatory response due to the recognition of the pathogen by the TLR. Another consequence of this recognition of the pathogen by TLR is that it enhances the capacity of the cells to kill microbes. It also augments its antigen presentation capacity. Antigen presentation refers to the fact that these foreign cells or foreign molecules which are antigenic in nature, they have to be presented to the immune system by a phenomena known as antigen presentation. what is the difference between toll receptors and toll-like receptors? Because we have used these two words or two phrases so far. Toll-like receptors have different structures in the extracellular domain as compared to the toll receptors initially observed in the fruit fly. The A part of the figure shows the toll receptor, and you can see that the extracellular part structurally looks very different from the toll-like receptor. If you kind of examine, the toll receptor has a domain which is called ectodomain, and it has some well-defined structural components. The toll-like receptors have extracellular domains which are little less complex. As far as the cytoplasmic domain or the transmembrane region is concerned, both classes of the receptors are quite similar. Let us look at the ligands associated with the toll-like receptors and some of the cells. The first column mentions the receptor. The second column describes the corresponding ligand. The third column is showing the list of the microbes, the corresponding microbes on which these ligands are present. And the fourth one, of course, is talking about the cells, which are written in abbreviated forms, as these abbreviations are defined at the bottom of the table. MC stands for monocytes, MA stands for macrophages, DC stands for myeloid and plasmacytoid dendritic cells, PMN are polymorphonuclear cells, FDC are follicular dendritic cells. TLR2 can form heterodimers with TLR1 or TLR6 with triacyl proteins and diacyl proteins as ligands. TLR3, TLR7, TLR8, TLR9 are intracellular TLRs found on endosomes. TLR10 is also known as a CD290. This has also been reported. Its ligands are less understood, they are not so well identified, but what is known as that its cellular distribution is on the plasmacytoid dendritic cells. TLRs have extracellular domain which is characterized by the richness in leucine rich repeats. They also have a cytoplasmic domain, and that is the domain which of course is involved in the signal transduction process. These germline coded molecules detect invading pathogens and trigger expression of cytokines. We had already mentioned that. Let us also add to that they also trigger expression of some other co-stimulating molecules. The best characterized member of this class is TLR4. Apart from lipopolysaccharides, which is shown here as the ligand corresponding to TLR4, it in fact also binds to the host proteins during damage, infection and inflammation like HSP60 and a fibronectin variant. Let us now look at what we refer to as a mannose receptor. Mannose receptor is also called CD206. It's a calcium dependent 180 kilodalton integral protein, has 8 carbohydrate lectin domains which recognizes mannose or fucosyl recognition patterns on pathogens which enter normally through mucosal surfaces. The mannose receptor also has a terminal lectin domain which recognizes sulfated carbohydrate groups. Macrophages, which are distributed throughout the body, endothelial cells and dendritic cells have these mannose receptors. In a way, mannose receptor link the innate immune response with acquired immunity, as the pathogens, after being recognized by these mannose receptor, are internalized, after which they are degraded in endosomes and displayed on antigen presenting cells. All this will be, of course, clearer once we start discussing acquired immunity in detail. This receptor or this class of receptors also binds host proteins such as myeloperoxidase, lysosomal hydrolases, and some hormones. Here are some examples of few other important receptors. The cells involved in the innate immunity display a very wide range of receptors with corresponding recognition of wide-ranging patterns on the cell surfaces of the pathogens. That makes sense because the cell surfaces of different pathogens are going to have a different composition of the ligands. They will also have a different distribution pattern. Having these variety of receptors with different specificities ensures that no matter whatever is the site of infection, whatever is the nature of infection, innate immunity initiates the immune response. As we have indicated earlier, it is the innate immunity which evolved first. So, in itself, it actually is a very versatile kind of a system in itself and tries to take care of all kinds of infections. So far, what we have talked about are the pattern recognition receptors which are present as integral proteins on the cell surface as a part of the biomembranes of the host. There are various pattern recognition receptors which are soluble in nature. Many soluble proteins recognize what are known as a pathogen associated molecular patterns. These are, in the abbreviated form, are called PMPs. These are mostly carbohydrate recognizing proteins and they are also part of the innate immunity. Lectins are one of the most important class of the carbohydrate recognition molecules, and they recognize carbohydrate whether they are present as free or as a part of the other more complex molecules. Later on, it has emerged that lectins are in fact a part of the broader class of molecules which are called carbohydrate recognizing proteins. Recognition of the carbohydrate molecules constitute a very important part of the molecular recognition processes in biology, and these PMPs illustrate this as they are very important example of this fact. Some of these PMPs which are shown here, collectins, ficolins and galactins are some of them. Collectins belongs to the collagen containing C-type lectin superfamily. Ficolins have a collagen domain as well as fibrinogen domain. Essentially, these recognize N-acetyl glucosamine and activate complement pathway, which we mentioned as another part of the innate immunity. Galactins bind to the glycans and directly kill bacteria. We have talked of complement system couple of times before. Let us briefly look at it. Complement system consists of 20 soluble glycoproteins which are constitutively present in blood and some other body fluids. Many of these are produced by hepatocytes and monocytes. On being triggered, a cascade of reactions occur which enables protection against pathogens through inflammation, enhanced phagocytosis, and lysis of pathogenic cells. The triggering can occur through three different pathways as shown. Complementary systems and these different pathways will be discussed in more detail later on. So these acute phase proteins include C-reactive proteins, complement components, MBL, metal binding proteins, and protease inhibitors. These are mostly produced by liver, either de novo, for example CRP, or their low level increases rapidly. An example of this is fibrinogen. These are particularly important in defense against bacteria and protozoa. Activated macrophages and NK cells release cytokines, IL-6 being very important, and this leads to hepatocytes responding by producing acute phase proteins. Overall, acute phase proteins limit tissue damage by microbial fractions, trauma, malignancy, diseases like rheumatoid arthritis. These proteins are important in tissue repair as well. This table shows examples of the various acute phase proteins. Please note that complement components are also considered part of these acute phase proteins. These various acute phase proteins occur as in the form of similar pentagonal subunit associations, and these acute phase proteins which have this kind of a structure are classified as pentatrexines. Examples are CRP, SSA, and SAP. All pentatrexines have another common feature, that they are calcium ion dependent carbohydrate binding proteins. Let us also devote 1 minute more on talking about cytokines. What is mentioned as their features or the classes, it is better not to take the above as a classification, but more like definition of terms, as there is some overlap among these. For example, interferons are variously referred to as lymphokines or cytokines. Among cytokines, interleukins are very important. 35 interleukins have been described. Some are better characterized than others. Some are also produced by lymphocytes, and in those cases they are called lymphokines. These can be placed in 4 families which are mentioned here, and this classification is based upon their structural similarities. Like other cytokines, these have diverse functions and highlight the overlap between innate and acquired immunity. Here are examples of some of the chemokines. More than 50 have been identified so far. These are 8 to 10 kilodalton peptides, largely responsible for chemotaxis of lymphocytes, monocytes, and other neutrophils. These have conserved cysteine residues, and their position and amino acid sequences form the basis of the 4 classes of chemokines. Receptors for chemokines are also integral membrane proteins, and the signal transduction operates via the mediation of the G protein. So these molecules activate and direct effector cells to the infection or tissue damage sites. These also control leukocytes migration in the tissues. Interferons constitute an important example of the cytokines. These proteins protect against viral infections. Many texts prefer treating IFN alpha and IFN beta as a single class because they actually have the same receptors. Interferons from the activated cells or virally infected cells bind to receptors on the nearby cells, which in turn produce antiviral proteins. Antiviral proteins limit viral replication by either blocking protein synthesis initiation or cause messenger RNA degradation of viral proteins. Here are some other important cytokines which are mentioned here: migration inhibition factor, tumor necrosis factor TNF and lymphotoxin, transforming growth factor beta, and finally CSFs, which include the granulocyte, monocyte, granulocyte CSF and monocyte CSF. They are briefly, their functions etc. are indicated in this slide. Apart from the NK cells which we talked about, phagocytes, which consist of the neutrophils and the macrophages, constitute other cells of the innate immune system. The other cells which are part of the innate immune system are mast cells and basophils, dendritic cells. There are some other cells which also have a role in innate immunity. For example, erythrocytes bind and remove small immune complexes. Eosinophils, which are granular leukocytes which release toxic proteins which kill parasites. Platelets, which release mediators that activate complement cells, and epithelial cells, whose role in releasing AMPs has already been mentioned. So, students, let us now summarize what we have learnt in this module. We have learnt innate immunity and acquired immunity are two kinds of immune responses. We have learnt that innate immunity is mostly non-specific and does not depend upon previous encounter. Innate response is mediated by soluble factors as well as cells. We also learnt that the pattern recognition receptors recognize pathogen associated molecular patterns. In this module, we have discussed the cells involved in innate immunity less extensively. The reason for this is that immune response is a complex interplay of various cells and soluble factors. The complete function of cells involved in innate immunity are better appreciated when we have developed a better background of some aspects of the immune system. So let us treat this as sort of introduction to the cast. Their roles in the drama will follow. Thank you.